Jorge Quiroz, who is from NMD Pharma, and he's going to talk to us about a chloride channel inhibitor.
Good morning. Thank you for the invitation. At NMD Pharma, we're very proud of the work that we have been doing so far, and wanted to share with you what the implications are for the molecule that we're developing in the clinic in CMT disease. There's a pointer. Excellent. As a general statement, we're developing a CLC1 channel inhibitor. It's a chloride channel type 1 inhibitor to increase muscle strength and function in neuromuscular diseases. And we have demonstrated already some clinical benefit with this molecule in a proof of mechanism study in myasthenia gravis. And we are using a set of biomarkers that are translatable from the preclinical studies into the clinic that I will share with you today. And from there, I will take you in what is coming moving forward for CMT. The chloride channel inhibition is a mechanism that you can find in the neuromuscular junction, depicted here. As you know, the main excitatory neurotransmitter is acetylcholine receptor and the activation by acetylcholine. And the main inhibitory one is the chloride 1 channel that is only present in striatal muscles. It's not present in the heart, for example. So what we have been developing is an inhibitor of this chloride channel 1. So it's an inhibitor of the inhibition, which as a result may improve the probability of fighting for the sarcolemma and therefore increase muscle strength and function. We have already a proof of mechanism study in patients with myasthenia gravis. We published this earlier this year in Science Translational Medicine, so I invite you to take a look on the translational work that was done to get into the myasthenia gravis indication. And we're doing and following some similar path for the treatment of the CMT disease, and I will walk you through that now. Of course, this mechanism of action may be applicable to a series of conditions because in a way, acting on the neuromuscular junction and on the sarcolemma side of the neuromuscular junction fundamentally, it is agnostic of what is happening and producing the disease. So we believe that may be utilized in presynaptic disorders and on their SMA and Charcot-Marie-Tooth disease, for example, but also in postsynaptic conditions like in myasthenia gravis, and from there the trial that I have mentioned to you. And again, just to repeat, the idea is that the inhibition of the chloride 1 channel will produce an increase in the neurotransmission. For any quanta of acetylcholine, you will have more power in producing muscular contraction postsynaptically. And for that, what we have been doing is utilizing the biomarkers of electromyography that have been quite translatable from what has been observed in the preclinical animals and animal models that we have utilized to develop the compound into the clinic. So as you know, and maybe even more familiar than what I am, we have been utilizing repetitive nerve stimulation, and with that measuring the CMMP, the amplitude modulation that happens when you repeatedly stimulate nerve and you measure in the muscle the amplitude. But also in the single fiber EMG, we can also quantify the level of jitter, which is the delay between two impulses, or the block, which is the absence of the second impulse after the first impulse is stimulated artificially or voluntarily by the patient. And on that biomarker, we have been building our strategy about the neuromuscular diseases. Let me show you a bit about that. I will not stop in Charcot-Marie-Tooth and the different types. I think it has been discussed already. To do this, we started with an observational study in patients, similar to what the colleague before was mentioning as their next step. We wanted to see if in patients we were able to observe these neuromuscular junction deficits that we know that are reversible with the CLC1 inhibitor that we have in the company. So for that, we collected information from CMT patients type 1 and type 2, and we compared with age-matched controls in healthy volunteers. and we investigated the association between the dysfunction in the neurotransmitter activity, on the neuromuscular junction activity and function, with the symptom severity. And the idea, it was twofold. One, to evaluate how these biomarkers in the preclinic translate into the clinical expression in patients with Charcot-Marie-Tooth, and second, to understand how these endpoints could be utilized later on in a study that is now ongoing, I will show you as well, in the disease. So for this, in this study, it's called Established. We're soon to publish. I think it's already approved or not? Not yet, but it's on review, under review. We have this prospective observational study where we have the two groups that I mentioned, the patients with CMT disease and the healthy controls. The healthy controls were age-matched, and we did measures of electromyography at the time point 0, and then the patients came 4 times in total, 3 times more, after that first observation to understand the variability of the measurements from the electromyology standpoint and also from the clinical standpoint. This study was conducted in 2 sites, one in the US with Dr. Dave Arnold at Ohio State when he was there, and the other one in Aarhus, which is the city where the company it is headquartered in Denmark. And there the Professor Henning Andersen, who is a very close collaborator and great colleague that we're working with as David is. And these are the findings from that study. And in my mind, I think in our minds, very interesting results. I think that the most fundamental one is we found, as expected, that there is neuromuscular transmission deficits in Charcot-Marie-Tooth, independently of the fact that this is demyelinating disease or if it is an axonal degeneration disease. We find that in CMT1 and 2, we observe jitter and blocking in a percentage or proportion that is much larger than the observing healthy volunteers. Healthy volunteers do not have jitter or blocking, or if they have, it's very minimal, very minimal. And you can see in green the patients compare in a high proportion of jitter and a high proportion of blocking against the healthy volunteers. Even more, the sample size didn't allow us to reach the conclusion, but the CMT2 patients are the one here on the top versus the CMT1 patients. It may have been that our CMT2 patient population was a bit more severe than the CMT1. We don't have enough data to make that distinction because of the sample size. But importantly, not only we detected the signal was failing, as is expected, if the neuron is not firing well, the neuromuscular junction, therefore, is also dysfunctional. We found that the jitter and blocking was very nicely associated. Look at the Pearson correlation numbers here, 0.8, 0.9, 6, etc., with the performance in their clinical test. And we did a myriad of them. So here they're all listed from ankle dorsiflexion, which is manual, to 10-minute walks or 7-minute walk tests, etc. And the correlations are relatively respectable. And even more, these are not random findings, at least in our eyes, because you can see that the p-values, although not pre-specified, are quite eloquent on telling us that these findings most likely were not due by chance. So again, as we discussed, the correlation exists there, and we also wanted to understand how well tolerated these tests would have been when performed in a repetitive basis with the patients coming into the clinic. And the tolerability was beyond our expectations. The patients were very collaborative, of course. A confounding factor is that these are the patients that wanted to collaborate with the research. This is a phase 0 study, and therefore there is no benefit. So maybe there is an ascertainment bias on who came to help us. However, you can see that the tests were well tolerated, and the reliability in the intra-center and inter-center was quite high. So with this information, we think that we can tap into these endpoints to develop a phase 2 study that is now ongoing. With the results from this phase 0 study, the team decided to try a tool compound, not the compound that is in the clinic, first, in preclinical animal models of the disease. So here I'm presenting one of them. This is the CMT2D model, where first it was observed that the CLC1 inhibition did improve, accomplish the function, did improve the conductance of the study in those membranes. The second is that when you stimulate the cells and you increase the number of stimulus toward the right, you can see that the animals with the compound get closer to the wild type versus the animal models without the compound, and therefore there is some sort of normalization with this tool compound. It's not the compound that we move in the clinic, so the potency is not comparable, but conceptually it's in the direction that we wanted to achieve. And thirdly, that these effects that are electrophysiological, if you wish, were also showing an improve in the measured force, and the sample size being very small, but the stimulation frequencies of 12, 30, or 80 hertz, the percentage of recovery was very important for the animals treated with the compound versus the wild type. So I'll summarize now. We have conducted a phase 0 study where we showed that the component of neuromuscular dysfunction is present in patients with CMT disease type 1 and type 2. We showed with a tool compound in an animal models of CMT2D that those deficits are reversible. So with that, we decided to move into the clinic where we are at now with a phase 2A study, a proof of concept study, where we are evaluating the compound that we already tried in the myasthenia gravis study that I mentioned before. It's called NMD670 in patients with Charcot-Marie-Tooth, where the primary efficacy endpoints has been pre-established, and I will tell you a bit about that, and where we're going to try to understand the safety and tolerability of NMD670 in this 2 patient population. The study is very simple. It's a parallel arm study where patients are going to be randomized to placebo or drug. The dose currently available, that a maximum dose that we can dose patients is 400 milligrams. We do it BID at 8 a.m. and at 2 p.m. to give coverage during the entire daytime in terms of increasing the muscle strength. We know that based on our prior experience with patients and healthy volunteers. We have done around 70 healthy volunteers with this drug so far, and we have a study ongoing in SMA, most likely around 15 patients so far, maybe 20. So we do have some experience on how to dose the compound and how much coverage the patients may have, and also we model this before getting into the clinic, of course. And after that, with 21 days of treatments, we will follow the patients for 7 days to understand what reversibility of the effects is observed if there is one improvement in efficacy. This study will enroll patients type 1 and type 2, but of course we're trying to enrich the patients type 2 to get valid information, so we learn from our experience on the observational study. We're trying to get more patients, hopefully around 30% of the population, so 12 to 24 patients in each, in the total, 12 in each gram for the study with having the CMT type 2 disease. And we are also including patients based on the performance of 6-minute walk test. And if there is time for the discussion, we can talk a little about that and why that would be a good measurement or not. And we are utilizing, we are collaborating with 20 sites in Europe and in the US, and I will tell you a bit more about the sites in the US in a minute. Let me go here a bit quicker. I'm sorry. So the main inclusion criteria for this study is patients 18 to 70 years old. This has to do with safety, with any limitation to the effect that is expected by the drug at that age. The 6-minute walk test needs to be into one standard deviation from what we define the norm is for the gender and the age of the patient. We have been able to, with the CMT Research Foundation, collaborate on trying to find out what is the normal performance of patients, what is the norm performance of patients with CMT, and based on that data, we have been able to predict and understand better what that norm would look like. They need to have some deficit in dorsiflexion, so the score can be 4 or less. And body mass index, again, for safety reasons, there is a limit at the 35 BMI. And the exclusion criteria, perhaps the most important, is that the patients in these proof of concept studies cannot be utilizing as AFOs. The orthopedic inserts are allowed, but other AFOs are not, and we know that a significant proportion of patients are in AFOs. But for these proof of concept studies and for the homogeneity of the population, we have decided not to do that after an extensive research that we did on the utilization of AFOs. and also severe deformities that, or contractures that may impair the patients to perform the clinical measurements would also serve as an exclusion criteria. The primary endpoint of the study is the efficacy of the NMD670, and we're going to be measuring 6 minutes walk test and comparing the delta between baseline and the endpoint at 21 days, and comparing the placebo and the active group in drug. But we're also measuring 10 minutes walk run and the time up and go that you may be familiar with, in addition of the CMT form that includes a couple of these, and many other neuromuscular function tests. We're also measuring fatigue that will be calculated from the 6-minute walk test. We're also measuring OANLS, CMT high to understand how the patients score themselves, and SF36. And of course, we have a subset of sites that are performing single fiber EMG to understand how the GTRM blocking are affected or not with the NMD670, the CLC1 channel inhibitor, and if those changes are correlated to the efficacy observed in those patients. And of course, safety is always the other important endpoint that will be collected in the study. I mentioned before that we are collaborating with 20 sites in the US and Europe, and this is the list of the US sites. Not all of these sites are already up and running, but this is a close, perhaps, prediction of what may be happening in the next 3 or 4 months. And the sites are here. We are listing the sites in the clinicaltrials.gov. You can find the trial there and the details of the design, perhaps less detailed than this one, but it's still relevant for you to derive patients that you think that may want to participate in the study and help us to understand if this drug is efficacious in the group. So with this, I'll finish, and of course, this is a work of a lot of people. My team, which is the clinical operations and regulatory team, has been working furiously to open the study that now is up and running. So with that, I finish, and I'll leave it to the coordinator for the next session. Thank you so much.
Okay, we have time for a couple questions. Got one over here.
Thank you very much. Cleobas is my name. So I have several questions, but I will limit to two. The one at the preclinical level, have you looked at the nerve morphology in this CMT 2D mice, or just the neuromuscular junction electrophysiology?
Yeah. So let me say it differently. The CLC1 inhibition does produce effects in the presynaptic portion of the neuromuscular junction. And I don't know how much we have disclosed those results, but that work is ongoing, and I can tell you from the generality that tend to be positive.
In terms of the clinical study, I mean, it's striking that you look only 7 days after treatment. So are you looking for a symptomatic treatment? Is it for a temporary effect? For example, like we use pyridostigmine in myasthenia because...
No, that's a great question.
And what about long-term effects if you keep stimulating the muscle? Because chloride channel dysfunction we know is associated with myotonia. So that could lead to prolonged muscle activity and prolonged stiffness.
Yeah, 2 questions there. So thank you. That's a great question, actually. On the duration. Of course, we want to dose patients for a longer period of time, but in drug development, you can dose for as many days. You have demonstrated that the toxicology studies are clean to the agencies where you are conducting the study. So we have a 4-week tox study that allows us to dose up to 4 weeks from there to 21 days. Now we have completed a 3-month toxicology study, but that was not completed by the time that we submitted the application, so that a change may happen there to continue dosing the patients. If it is symptomatic or not, I will say that the drug improved muscle function. It is not expected that the mechanism of action will target the axonal degeneration or the remineralization. However, in the studies that I mentioned before, we are seeing an effect in the presynaptic neuron that may be relevant for the disease. That we don't know yet. So that to answer the first part. And the second had to do with myotonia. Yes. So we did observe myotonia in healthy volunteers at very high dose, much higher than that. I can tell you, it was 1,200 milligrams and 1600 milligrams one dose. Patients did have myotonia that was of moderate intensity. Not patients, healthy volunteers. When we went into patients at the same doses in myasthenia gravis, we did not observe myotonia. And in all cases, most likely myotonia will be Cmax related. So basically, when the concentration in plasma is very high, that's when you can have the myotonia. These doses are 3 times lower than the one that was observed in the healthy volunteers. So it is expected that no myotonia will occur in patients with maybe less sensible to this mechanism of action because they have a failing neuromuscular junction. But in addition, we have the safety factor protection that will allow us not to get there. We're following this very closely, and I didn't tell you in the detail, but I disclosed there all the things that we're following. One of them is myotonia as a secondary adverse event. That's a great question. Thank you.
Okay.